Welcome to the Basics lecture on translation. In our previous video, we covered transcription, making of mRNA. Now we're going to take that mRNA, take it out of the nucleus and into the cytoplasm, and translate it into a polypeptide. Every 3 bases in an mRNA strand is called a codon. They form 3-letter words that code for the 20 different amino acids that make up proteins. A table like this one can help you understand and apply codons. For instance, this first codon is a UGG. To figure out what amino acid is associated with that codon, we find the U and then go over to the G, which brings us to this box here, and we find that UGG codes for tryptophan, TRP abbreviation. Take a moment to figure out the other 3 amino acids shown here, and then compare your answers to that in the book in Chapter 17. tRNA molecules are the critical connector between mRNA and its amino acids. This image shows a realistic 3D image of tRNA and a cartoon that we'll use from here on out on the right. These molecules are made of RNA, but they leave the nucleus and are not translated into protein. Instead, they have anticodon binding sites on the bottom that matches the codon on the mRNA, and on top is a site that binds to the specific amino acids. The anticodon on the base of a tRNA contains bases that correspond to the codon on the mRNA. If this first codon on the mRNA is GAU, read 5' to 3', then the anticodon would be CUA. Fill in the anticodons for the remaining 2 mRNA codons. tRNA molecules are reused and need to be bound to an amino acid before each use. There are 20 different enzymes called aminoacyltRNAs in the cell, each one of which is shaped so that it only binds to one type of amino acid and one tRNA anticodon. The energy released from hydrolyzing 2 phosphates from ATP is used to attach the amino acid to an active site on this enzyme, and then the appropriate tRNA for that amino acid binds to the remaining active site, which removes the adenosine monophosphate, gets that out of the way, and then a peptide bond forms between the tRNA molecule and the amino acid. The charged tRNA is then released from the enzyme and able to take that amino acid over to a ribosome. So now we know enough to put all the parts of translation together. Initiation begins when an mRNA binds to the small ribosomal subunit, shown here, and an initiator tRNA binds to the start codon. You can see that there's some of the mRNA that's not utilized to build an amino acid. We've heard about the cap that we saw in the previous lecture. The start codon for every protein is always AUG, which means that the first amino acid on every protein is always a methionine. The large subunit then attaches to the complex, and a GTP is used to hold them together. Elongation has about 4 steps that you need to worry about. A new tRNA binds onto its codon on an open spot on the ribosome. The polypeptide chain is transferred to the new amino acid via a peptide bond. These tRNA molecules and their mRNA advance in the ribosome so that they fill the second 2 places in the ribosome. And the empty tRNA that had the polypeptide removed from it falls off, and the new tRNA is now at the center position in the ribosome, ready to receive the next tRNA. You don't need to remember that GTP is hydrolyzed at specific locations in this process, but you should remember that the process requires cellular energy. The polypeptide terminates when the ribosome reaches the stop codon that is at the end of all mRNA molecules. The stop codon, which is either UAG or UAA or UGA, binds to a release factor protein instead of a tRNA molecule. This blocks further translation. You can see that it doesn't have an amino acid on it, and the mRNA and polypeptide are released from the ribosome and the subunits fall apart. So far, we've only shown a single ribosome, but most strands of mRNA have multiple ribosomes attached, creating a polysome. The first ribosome binds to the 5' end and begins translation, moving down the strand towards the 3' end. Once this first ribosome has moved down the mRNA several codons, there's space available at the start codon, and a new ribosome can then bind on top. So multiple ribosomes make their way down the mRNA molecule and allow multiple polypeptides to be made from a single strand of mRNA before the mRNA degrades in the cell. Now that you understand the basics of making a polypeptide, let's look at what happens if there's an incorrect nucleotide base pair and how that affects the mRNA and the protein. Here is a normal wild-type strand of DNA. The top strand is the template. It codes for the mRNA, which is then used to make the protein. So far, that should seem like what we've just covered. If there is a single base pair substitution, perhaps due to an error in DNA replication, you might see that in that the G here being changed to an A. This can affect or not affect the resulting amino acid. In this particular case, this change creates a uracil instead of a cytosine, but because both GGC and GGU code for glycine, there is no effect in the primary sequence of amino acids. This is called a silent mutation. You see no change in the polypeptide due to the change in DNA. A missense substitution, however, changes the resulting amino acid, which might affect protein function. In this example, the substitution changes the codon from GCC to an AGC, which changes the amino acid from a glycine to a serine. This is called a missense substitution. The change in structure might strongly affect the resulting protein, and it might not. It varies from protein to protein. In this case, the substitution from thymine to adenine, shown earlier on in the sequence, creates a uracil on the mRNA. In this case, this generates a stop codon where there wasn't one before. So it's not causing a different amino acid to be present, but instead terminates the polypeptide too soon. This is called a nonsense mutation, which means it stops the polypeptide translation. Now, sometimes mutations are not called by substitutions, but by actual insertions of additional base pairs or deletions. In this first example, a single nucleotide insertion, shown here, causes a frame shift, which means a single amino acid, or a single base pair, switches the codons afterwards by moving each codon one base pair forward. In this particular example, the insertion created an early stop codon, so it's a nonsense mutation. A deletion can also occur that removes a base pair. This again causes a frame shift. All the codons are now different. And in this case, it caused a change or a missense mutation rather than a stop codon change. Now, if a full codon is added or lost, there is no frame shift, assuming that it was a codon itself and not just three random base pairs. The addition or loss of amino acid may or may not affect protein folding and protein function. Here, the example removes an entire bit of DNA, which removes one single codon, and just the lysine amino acid that was seen in the normal cell is now, or the normal protein is now gone altogether. A mutation is defined as any change to the genetic code, whether or not it causes a change in the transcribed protein or even any problem in the organism. We've just finished looking at mutations caused to small changes in base pairs. Earlier we saw indications of large-scale mutations with large chunks of differences in chromosomes. And lastly, here are changes in chromosome number. Take a minute and look up the definitions to these in your book's glossary or in Chapter 15 so that you have an idea of the full range of mutations possible in the cell. This image is a nice summary of all the major steps in protein synthesis, transcription, RNA processing, amino acid activation, and translation. Make sure you can describe all these steps. That's our video for today. Thanks for watching.